Um, Thank you for attending part one of our mini summer research series, Progress on Gene Therapy for Classical Homocystinuria, also known as Cystathionine Beta-Synthase. I am Danae Barkey, an adult homocystinuria patient and the Executive Director of HDU Network America. I will be your host for today. I'd like to introduce our honored speaker of today, Dr. Warren Kruger. During his postdoctoral work at Stanford in the Human Genetics Lab of Dr. David Cox, he developed a yeast functional assay to study the functional effects of the human cystathionine beta-synthase mutations. Since 1995, he has been a researcher at the Fox Chase Cancer Center in Philadelphia, where he is currently a full professor in the molecular therapeutics program. Dr. Kruger's work focuses on the role of dysregulated methionine metabolism in the human disease. His lab has made numerous contributions to the study of CBS deficiency, including the clone of the gene, creation of functional models of yeast and mice, and key discoveries in the regulation of enzymes. He enjoys scientific collaboration, and the mice created in his lab are used to study homocysteine-related disease throughout the world. Thank you, Dr. Kruger, for joining us today.
Thank you. And it's a pleasure to be here and to get to talk a little bit with some actual patients with homocystinuria. So I am going to get right into my presentation here. So just bear with me. Share screen. Share. And then I'm going to make it big. Oops, it's not what I wanted to do. And I need to go back to the beginning, which is a mistake. There we go. So the title of my talk today is Gene Therapy for Classical Homocystinuria. And classical homocystinuria, the more proper name is cystathionine beta-synthase deficiency. And I'm a professor at the Fox Chase Cancer Center in Philadelphia. Half my lab works on cancer-related stuff, but for historical reasons, I've been working in the field of methionine metabolism and CBS in particular for the last 25 years. So, or actually longer, boy, I'm getting old, almost 30 years now. And this talk, I'm gonna try and keep it simple, but yet still show you a little bit of science too. So you're probably all aware, classical homocystinuria or cystathionine beta-synthase deficiency is an inborn error of metabolism, and it's characterized by extremely elevated levels of a molecule called total homocysteine and methionine in your blood. Homocysteine is what we call an intermediary amino acid. It's not found in protein, but it can make other amino acids. People with CBS deficiency have a variety of issues that can affect them as they go through life, including problems with the vascular system, specifically blood clots, the skeletal system, osteoporosis is very common, central nervous system, certain behavioral and mental challenges, and then the visual system oftentimes is what brings CBS patients to the clinic initially as dislocated lenses. As you all are aware, if you control your total homocysteine levels, you can greatly lessen the likelihood of having a complication from the disease. We'll talk about how you do that in a minute. And the other thing to remember is it's a recessive disorder. So you have 2 copies of every gene in your body, and you need 2 bad copies to have CBS deficiency, one from mom and one from dad. This slide shows in the middle, I don't know if you can see my pointer. Can you see my pointer, Dan? I hope so.
Yes.
Oh, okay. So this is homocysteine, and this is a chemical structure. And O is oxygen, H is hydrogen, N is nitrogen, and S is sulfur. And so homocysteine looks like this, and it's got this sulfur group at the end. And it's derived from this molecule, methionine. And you'll see methionine looks just like it, except the sulfur has this methyl group attached, whereas this one only has a hydrogen. The other related molecule is called cysteine, and cysteine also has a sulfur group, but it's missing one carbon chain. That's why it's one little jig shorter than homocysteine. And the important point that I just want to make here is that methionine is essential in the human diet. And the reason it's essential is you see how this sulfur group here is attached to a carbon here, and this is a carbon here. Humans can't attach sulfur to carbon. So the only way we can have sulfur and carbon attached is by getting it from our diet. And the source of most of it comes from methionine, and we can get some from cysteine. Okay. The other thing I want to point out in this slide is that in a normal individual, homocysteine levels in the blood would be about 7.5 micromolar. And in a CBS deficient patient, that number is much, much higher, over 10-fold higher, typically over 100 micromolar. They can be even higher than this. They can go up to 200 micromolar. These numbers came from a doctor, Sally Stabler at University of Colorado at Denver, from a bunch of patients she's seen. So this is an average number. The other thing that happens in CBS deficient patients is that their methionine levels go up as well. But for reasons that I won't go into today, we know that it's having high levels of homocysteine is the bad player. The methionine we don't think is doing too much. One other feature is cysteine levels tend to go down in homocysteinuric patients. The importance of that is not fully understood. So those are the molecules, oops. And this is the metabolic pathway, how all these things are related to each other. So you get that methionine I told you about from your diet, and it's used for two different things. It can be used to make protein, which is obviously important. We all have to make new proteins. But the other thing that methionine does is it gets converted into a molecule called S-adenosylmethionine. And S-adenosylmethionine plays an important role in our bodies in that it donates methyl groups to a variety of different chemical reactions. So putting on a CH3 on a molecule, it turns out that's something the body does all the time. And in fact, there are over 200 different enzymes or chemical reactions that utilize SAM to put these methyl groups onto different molecules. When you get rid of that methyl group, you make something called S-adenosyl homocysteine, and that then gets converted to homocysteine. And it's this homocysteine that we want to get rid of. And there are two ways to get rid of it. One way is we can convert it back to methionine. And there are actually two different chemical reactions that do that. One reaction requires a vitamin, vitamin B12, and another vitamin, folic acid, which is used to make folates. And what happens is it takes the methyl group from the folate and it puts it onto the homocysteine, and you get methionine. And that reaction is found in most of the tissues in your body. There's another reaction that's only found in the liver where homocysteine gets converted to methionine by giving it betaine. And I think some of you may be familiar with taking betaine to help control your homocysteine levels. And that's how it works, is it helps stimulate this reaction. But there's another way to get rid of homocysteine. And in fact, the most important way is to get rid of it by converting it to cystathionine and ultimately to cysteine. And this is where CBS is important. So CBS, what it does is it takes homocysteine and another amino acid called serine, and it combines them together to form cystathionine. And CBS also uses a vitamin. This one used vitamin B12. This one uses a vitamin called B6 or pyridoxine. And then you get your cystathionine, and then cystathionine gets broken down to become cysteine. So the problem you have when you lack CBS is you build up this homocysteine, because normally a lot of homocysteine is converted down this pathway. Now, you might think, well, why don't I have a problem by not having enough cysteine? It's an amino acid. It's used to make protein. It's really important. And the reason is you tend to get enough cysteine in your diet to compensate for the lower levels of activity through the pathway. So as a scientist, I look at this pathway rather than as important for the synthesis of cysteine. It's really there to get rid of your homocysteine. Homocysteine is bad. One other thing is, and this is kind of technical, but if you ever get your homocysteine measured, they talk about total homocysteine. And the reason they do that is because homocysteine is actually found in a bunch of different chemical forms, because it turns out this sulfur is very reactive. So sometimes you get it just as what we call reduced homocysteine, but a lot of times it's oxidized. The sulfurs bind to each other and you get two molecules, and we call this homocysteine. And sometimes you even get things that are like homocysteine and cysteine, because cysteine also had a sulfur group. But most of your homocysteine is actually bound to protein in the blood and also in your cells, where it finds cysteine residues in protein and binds to them. And for reasons I won't go into, I actually think this is the form that is actually may cause a lot of problems, because I think it'll interfere with the function of these proteins. But the bottom line is we don't really understand how come high levels of homocysteine are toxic. And that's been the case, unfortunately, for a long time. It's a hard question to crack. So the cystathionine beta-synthase gene encodes a protein, an enzyme called cystathionine beta-synthase. This is a picture of it. We actually have, about 10 years ago, not even, 8 years ago, we were able to obtain what's called a crystal structure of the CBS molecule. It's slightly altered from the normal one, but it forms a dimer. So that means there's two molecules here, one that's in green and one that's in teal. And the CBS protein, it binds vitamin B6. That's this yellow thing here. This is where the actual reaction that takes homocysteine and serine and cooks them together takes place. But it also has this interesting molecule over here in red that's called a heme. It's the same heme that's found in hemoglobin. And it doesn't play a role in the chemistry. We don't exactly know what it does, but it's important for the function and the stability of the protein. And then the protein also binds S-adenosylmethionine, which actually regulates how the protein works. When this thing binds, the enzyme undergoes a change in its conformation so that it works better. One thing to note is that the gene is located on chromosome 21, and it's actually located in the region that is amplified in Down syndrome. So there's actually people are interested it seems like loss of CBS, that's homocystinuria. And some people think, well, if you have too much CBS, that can be bad too, and maybe contribute to Down syndrome. The protein is normally expressed in high levels in the liver, somewhat lower, but still high in the kidney, and low levels in all other tissues. As I said, it requires vitamin B6 to work, and it binds these two molecules. So that's just a little basic on what the protein and the enzyme look like. Now, the genetics are also pretty simple. It's recessive. You need 2 bad copies to have the disease. 1 bad copy means you are a carrier, but do not have symptoms, okay? So if you have 1 bad copy, you're basically fine. There are many different mutations that cause CBS deficiency, and some are associated with a person's ancestry and are found in certain populations. However, others are very rare and are only observed in a single patient from a single family. And that's because new mutations are occurring all the time. But some of the more common mutations are found in certain populations. So people of, for example, Irish heritage, there's a particular mutation that's frequent in those patients. And people who are from the country of Qatar, they have a very high rate of CBS deficiency, and they have their own specific mutation. That's because it originated in a tribe in Qatar. And I should say, overall, the frequency of CBS is somewhere around 1 in 100,000 births. Okay, so for every 100,000 babies that are born, you'll find 1 with CBS deficiency, at least in the West. Some mutations respond well to treatment with high-dose pyridoxine vitamin B6, while others do not. However, because individuals often have 2 different mutations, 1 in 1 copy, 1 in a different copy, a different one, it's not always able to predict which mutations are pyridoxine responsive or not. So generally, the physicians will give you pyridoxine as your first treatment and see how you do. And if you do very, very well, that might be all that you're treated with. But in general, only about maybe 1/4 to 1/3 of all patients respond well to high-dose pyridoxine by itself. People with pyridoxine responsive mutations do tend to do better clinically. And despite lots of research, including by yours truly, the mechanism is still a little murky on that. So, what is the current strategy? What do we do today? Well, we obviously give vitamin B6, that's easy. The most important mainstay is methionine restriction. So having a diet that's low in the amino acid methionine, because all homocysteine is made from methionine. So if you don't take in much methionine, you don't make much homocysteine. And usually, because methionine is found in protein, this translates to low protein diets, which can be difficult for patients. Another thing that's common, especially in European patients, is betaine supplementation. So they'll give patients a supplement with a lot of betaine in the hopes of driving their homocysteine towards methionine. And the effectiveness of all these treatments is highly variable and depends on good compliance. It depends in combination with whatever mutations you have. And it can be particularly challenging. So we want something better. And there are kind of 2 general strategies that I see as the future. So one is enzyme therapy. And this is actually something that has been developed by the late Jan Kraus, who was my competitor / collaborator for many, many years, who unfortunately died recently. And a lot of the work was done by a very talented research associate in his lab, Thomas Metchan. And Jan developed basically a genetically engineered CBS molecule that you could inject into the blood and will break down homocysteine. And that product, which is actually has a name now called pegtibatinase, is in human trials and is owned by a company called Travere Therapeutics. It requires IV injection, and I'm a little murky on this, but I think at least 2 times a week. In mice, it works really well. I don't know how the human trials are progressing, and I'm unable to comment about that. But I thought it's important that I mention that here, and you could, call Travere and find out if there are any openings in your area. My lab has been interested in recombinant adeno-associated virus, or RAAV-based gene therapy. And the advantage of gene therapy over enzyme therapy is pretty obvious. Here, what we're going to do is you have a bad gene, we're going to give you a good one. And in theory, it should last much, much longer, hopefully forever. And another advantage is we're actually going to express it in the tissue that has a lot of CBS, which is your liver. And hopefully that will help in some way. So how does gene therapy work? So basically what you do is you take a piece of DNA that encodes your gene of interest. In our case, it's the gene for Cystathionine Beta-Synthase. We're going to encapsulate it in a human virus. In this case, it's adeno-associated virus. And we're going to inject it into you. And what's going to happen is that virus is going to infect your liver, going to inject the DNA that it's carrying into your liver cell, and then your liver is going to start producing the protein, in this case CBS, that you're missing. So it's a very simple concept. I should mention, people have been talking about gene therapy since the 1990s. I remember as a graduate student, even as a graduate student, it was always, oh, this is going to revolutionize. It's taken a long time to get here, but it is finally here. And so far, the FDA has approved 2 different gene therapies using this AAV mechanism, one for retinal dystrophy, so they actually inject it into the eye, and the other is for spinal muscular atrophy. And there are ongoing trials with at least 20 different other gene therapy products that use AAV-based gene therapy. So this is happening, and it's happening quickly. So what does it take to get a gene therapy for CBS deficiency? And this is kind of my view, is the first is we have to design a viral vector using DNA technology. We can do that, and we've done that. You have to produce and harvest the recombinant virus. We can do that. And we have to test whether this thing actually works using a mouse model. And we've done that, and that'll be most of the data I show you today. The last 2 steps is you have to apply for an investigational new drug approval from the FDA. And we're working on that. But that costs a lot of money, and we're trying to find a corporate partner to help us with that. And then the last step is we do human clinical trials. So this just shows you a little bit more about the science behind how this works. So what we do is you have your vector, what we call your recombinant AAV vector, and this expresses your gene of interest between these 2 black repeats. These black repeats basically are DNA sequences that tell the cells that they should package this in the virus. And then in this white sequence between them is where we put the human CBS gene. And we take that piece of DNA and we transfect what we call a helper DNA that encodes all the proteins on the virus coat into a cell line. And then they produce the virus that you have to harvest, you have to purify, and then eventually you can put into people or mice. The one thing I want to point out is there's some key variables in the procedure. One is what we call the capsid subtype. So there are many different AAV subtypes, and each one has a different protein on their surface, and each AAV subtype has different tissue tropism. That means some like to infect blood, and some like to infect kidneys, and some like to infect livers. So you have to find the one that works best for you. And the other key variable is with the CBS gene, you have to tell it to be expressed in the tissue you want it to be in. And that's done by a little DNA sequence upstream of the gene that we call a promoter. And so we have to pick the right promoter. And you'll see why I'm discussing these two things, because sometimes you don't know right away what the right combination is. So when we initiated our study, which was actually funded by HCU Network America's founder, Margie McGlynn, this was something that Margie actually very much motivated me to do. She said, Warren, why don't you do something with gene therapy? I said, well, I don't know much about gene therapy. She said, here, talk to this guy at CHOP, he'll help you. So I just should have his name on here, but Philip Johnson was the guy at CHOP, and he suggested we try this system. So we use the AAV1 serotype and what's called the CMV promoter. Okay, so this is it's a strong viral promoter. And so we made this piece of DNA, it looked just like this, 7,696 base pairs long, and it's got the human CBS gene, the CMV promoter, it's got a way to end the gene, and then these, they're called interspersed repeats that allow the virus to get packaged. And then we sent this off to the viral vector core facility at Cincinnati's Children's Hospital. And a month later, they sent us a bunch of virus and we tested it. And so we tested it using our mouse model. And I want to just give you a little brief intro to that because it's important for everything we're going to do. So it turns out that mice that lack CBS activity, they're in worse shape than most patients are here because they all die by 1 month of age. And so to get around this problem, our lab made a mouse that's deleted for the mouse CBS gene, but contains a transgene, which is an extra bit of DNA that we put in from outside that encodes a mutant human CBS protein that has just a teeny little bit of activity, just enough to keep them alive. However, so they can survive, but they have severe CBS deficiency. And how severe? They have serum total homocysteine between 250 and 450 micromolar versus a normal mouse that's 3 to 7 micromolar. So these mice have pretty severe homocystinuria. And when you look at them, the most striking thing about them is they develop this age-related hair loss or alopecia. So at 120 days, we have 2 mice here, they're siblings. One has got 2 bad copies of CBS. This one only has 1 bad copy. And you can see the one with 2 bad copies, even at 120 days, is looking a little different than its sibling. Its hair is lighter, starting to lose hair on its head, and it's also smaller. And by 210 days, it looks really different. You know, it looks a little like Yoda, I always thought. So they have this alopecia phenotype. They're also much smaller, and that's mostly due to the reduced fat mass. They don't express an important gene for fat formation called SCD1. They also have osteoporosis, which is same as in humans. So they have decreased bone mass density. They have about a 20% decrease in their lifespan relative to their siblings. They have a lot of differences in how their genes are expressed in the liver, although for the most part, their livers function pretty well. And what's interesting is, just like in humans, if you take these mice and I put them on a diet where I only give them 1/8 the normal methionine levels that are found in mouse chow, they look totally fine. You can't distinguish them from their sibs. And that's work we've done throughout the years. So we took those mice and we injected this virus and we measured their homocysteine levels. So the way the experiment's done is we first measure the homocysteine before, and then we measure the homocysteine after, 7 days before the injection, 7 days after the injection. And we tried giving them a bunch of different concentrations of the virus. And this is PBS, this is a control. And we actually tried a bunch of different ways. We gave it to them by interperitoneal injection, by IV injection, and intramuscular injection. And the bottom line was that we saw a lowering of homocysteine when we gave a very high dose of virus, when we gave it by IP, but it wasn't very impressive. So they went from like 350 to 280. And this is a lot of virus, 8 times 10 to the 12th molecules of virus. But it was a hint. And when we sacrifice those mice and we harvest their livers, I know that sounds gross, but that's what we do. You euthanize the mice and you take their livers and then you measure the CBS activity in their liver. We were able to detect some increase in CBS activity, about a 4-fold increase in the mice that were treated with the highest dose. So this told us it was working a little bit. I should mention a normal mouse is 1,000 units. So this isn't working great. We only got 40 units, but it's working a little bit. So what you do in science when things work a little bit is you try and make them work better. And so we decided to do a second try. And this time we collaborated with a very skilled gene therapy expert at Cornell named Ron Crystal. And Ron suggested that rather than AAV1, we try a different AAV virus that has really good liver tropism called RH.11. And so we did that. And then we changed our construct, and instead of the CMV promoter, we used another promoter called a CAG promoter, which is, interestingly, a genetically engineered promoter, but it has really good liver expression. And so we made these 2 changes. And then we also changed where we produced the virus. We did it at his core facility at Cornell. So this is what this new vector looks like. Looks pretty similar, but different promoter and different AAV. And what we did is we got the virus and we tried a few different titers, different concentrations. So what this shows is we injected this much virus in 2 mice, this much in 2, this much in 2, this much in 1 or 2, either IP or IV. And then we let the mice go for either 21 days or 78 days. And the other thing I want to point out is these are really old mice. They're almost a year old. So they're totally bald and they look horrible. So we did that experiment and wow, we got this data and we were really impressed. So in the mice that we didn't inject, we had around 400 micromolar homocysteine out to 21 days. And the mice that we gave the highest dose to, which is 5.6 times 10 to the 11th, which if you remember is 10 times less than the amount we used in the first experiment. Look at what we're getting is homocysteine going almost to nothing, to normal levels here. And it appeared that the more, as we increased the dose, this is the lightest dose here, it didn't work as well as if we gave more. So we got what we call a dose dependence, which is good. And then we compared delivery methods, and the bottom line is we didn't see a difference whether we gave it by IV or IP injection. so we harvested livers from these mice, and wow, we saw incredible activities. In our control mice, we had 7 units of activity. And in the mice that we gave the highest dose to, we had 2,412 units, which was higher than our wild type control mice in this experiment. So in the liver, this thing worked great. Now in the kidney, it really didn't work at all. You can see that control mice have about 107 units of activity, the positive control mice. The negative control mice have 8, and these things are just like the negative controls. So that kind of told us 2 things. It says you can lower homocysteine a lot with just good liver expression, and that expressing CBS in the kidney is probably not that important. So we let this first experiment, some of the mice we let go all the way till 77 days. And as you can see, their homocysteine levels stayed really low the entire time. And at the end of 77 days, they had 1,643 units of activity. Again, even more than the wild type control mice. The other thing we noticed was that in the mice that we gave the virus to, their hair and whiskers started growing back. So these are control mice. So these mice just got the buffer. They didn't get the virus. And notice there is bald and they don't have any hair. And these mice, their hair is all starting to grow back. So we felt like they were getting better as well as they were lowering their homocysteine. So we then did a study where we followed the mice for 1 full year. And the other difference was we gave these mice, they were young. They were only 40 days old. So mice are weaned at about 30 days, and these are 10 days after weaning. We gave them their single dose of virus. And this is what happened over a year. The control mice, you can see, have very high homocysteine the whole time. The wild type mice have very low homocysteine the whole time. And the mice that we injected, and this is the percentage reduction from the untreated mice. You can see for the first, I'd say 119 days, they were totally almost like the wild type mice. There was a gradual creeping that occurred as the time went on, but still a dramatic reduction. And one thing I should point out is that a year for a mice is 1/3 of its lifespan. A mice will live about 3 years if you really take good care of it. So this would be the equivalent of like a human for 25 or 30 years. So I think it looks pretty good. And here we're measuring the weight of the mice. You can see that the wild type mice put on weight with this curve up here. The untreated CBS deficient mice obviously put on weight much more slowly and are much smaller. And you can see that the treated mice start out like the mutant mice and they end up very close to the wild type mice. This is liver activity. Again, we saw lots of liver activity even after a year. This is protein. I don't know, I don't think we'll go into how to read Western blots, but the upshot is where you see all this dark stuff here, that means they have a lot of CBS protein. And when we measured at the end of the study, we did what we called DEXA, which is a way to measure bone mass density and fat mass. If women have, people with osteoporosis will have DEXA human studies. And you can see here that we see that the wild type mice have more bone mass than the mutant mice, but the treated mice look just like wild type. And that was true with the fat mass as well. Lean mass increased, but not quite as much as the other two. And then this is what they look like by the alopecia phenotype. You can see their hair, they're totally healthy. They're look pretty much indistinguishable from the control mice. And you can actually do microscopy and show that they have more hair follicles. Their dermal layer is much healthier. See how much thicker it is here than it is here. And their hair is much thicker. Their whiskers are much thicker. Everything about them looks just like the wild type mice. So in summary, the AAV gene therapy corrected basically most of the phenotypes in the IT78T mouse model of CBS deficiency. We saw the correction last at least 1 year with a single dose. And the amount we used, which would be the equivalent of 2.2 times 10 to the 13th virus per kilogram, is well below that associated with toxicity in some human trials. In humans, when you get above 10 to the 14th, you can have problems, but this level, there shouldn't be a problem. And where we're at right now is we're in serious negotiations with a couple different biotech companies, I can't tell you their names, to try and have a licensing agreement between Cornell, Fox Chase, and the company so that money can get underway to initiate the human trials. So with that, I just want to give some credit to where credit is due. This is me, a lot of the work in my lab has been done by a very talented research associate, Hyung-Ok Lee. Sapna Gupta and Lee Chun were instrumental in making the CBS mouse models, and Bei Ching has been a long time research associate in my lab as well. We had help from Phil Johnson initially at Children's Hospital of Philadelphia, Ron Crystal at Weill Cornell, Stephen Kaminsky, who runs the gene therapy core there, and this is where funding has come from. So with that, I'm more than happy to answer questions of any kind on CBS deficiency or other causes of homocystinuria, whatever you want, I'll do my best. So thank you.
So we have sorry, I'm going to turn back on my camera here. And I will remove, sorry, the spotlight. So we're both seen here. Okay, so we have a couple of questions in the chat here. And the first one was, in the year-long mouse study, how many treatments were given to each mouse?
One.
Just one. Okay.
Well, at 40 days, we injected a single treatment.
Okay. And then let's see here. Do you anticipate that over time, since that was just 1 year out, do you anticipate that those antibodies would stay consistent in the body for a longer projection?
It's not an antibody, it's an enzyme. But so the question is a good one, and we would anticipate that you would probably require a second dose at some point, in my opinion at least, only because the liver will turn over. Now, in a mouse, and this is a really hard estimate to come by, and I've been doing some literature searching, it seems like the half-life of a liver cell in a mouse is about 1 year. The question is, what is the half-life in a human? So does it scale to lifespan, or is it constantly 1 year? So a human obviously will live 80 years, right? Mm-hmm. So so there's this gets complicated, but in the aging community, it seems like aging is kind of one of these things that's hardwired. In other words, no matter how hard you try, you're not going to get a mouse to live 100 years. And so the reason for that could be because their what we call their stem cells, the cells that make new cells, they just wear out after a while. And in mice, that might be 3 years. In humans, it might be much longer. And one of the reasons that could be the case is that the turnover of new cells is much slower in humans. So that's an unknown, and I don't think we're going to really know the answer to that until we do human studies. Once we put them into humans, we'll be able to see how long the activity lasts for, and if there needs to be a starter dose, a second dose, we can think of clever ways to do that.
Okay, said,
So I hear there's some in the chat.
Yes. Did the gradual creeping back of the, it says HCS levels in the Cornell test suggest any changes to the treatment might be needed? Or was
so, as I said, based on the amount of enzyme activity we had, I thought that it was actually really promising. So if we want to get technical, so we can make a better CBS enzyme. So it turns out that CBS is a lousy enzyme. It's not engineered to be the most efficient that it can be. And one of the things we're thinking about is if we make a more efficient CBS molecule, we might be able to keep it low for longer. And let's say we could make the molecule 10 times more efficient. If we gave it at the same dosing level, then presumably we'd have more than enough CBS to keep, even if it does get lost over time, there'd be so much excess that you would keep homocysteine levels low.
Okay. And then there was a question about, was the diet between the mice the same, like in terms of the protein level? So
yes,
okay.
Yes. So these mice were all on what we call standard mouse chow. We didn't put them on low methionine chow. If we combine this with low methionine chow, it'd be, and what we found is that if we put the mice on a low methionine diet, they look actually pretty much identical to the mice on gene therapy. So we think this will work as well as low methionine. And these mice are only getting 1/8 their normal methionine doses per day on their low methionine diet, which is, my guess is, much better than a human low protein diet is. You're probably doing more than one. How many protein grams did you say? Do you get a
Myself, I do 20. It's a higher tolerance for
20 grams a day. And what would normal be? Do you know what a normal?
I think most patients are somewhere under, like, between 10 and 15.
No, but I mean, like what? How much protein would I eat a day?
Oh, it depends on the kind of diet, but most Americans are eating well above. The suggestion is between 60 and 80, depending if you're male or female. But
right. So at best, you're probably cutting, you're at 25%. We can put the mice down to half that. Okay. That's what I'm saying, is that this will work really well.
Okay. Assuming those talks with those biotech companies go well, how long before we start to see clinical trials? And is there work that still needs to be done? Before?
So that's a good question. I would say, based on my talks with Ron, who has had experience with this, he feels that we could certainly, if the funding was available, he thinks we could get an IND in place and a trial protocol approved within 2 years. And that we could have the phase 1/2 going at that period of time.
So it's still a while off before, assuming every clinical trial phase, which we have a webinar talking about the pipeline there, but next after this. But assuming that all goes according to plan, it's still at least several years out, assuming everything is successful in the human.
Correct. Although I would say for, people that were interested, once the clinical trial opened up, that this would be one that you might you might consider, because
Yeah.
it's but yeah, I'll be honest with you, the big expense here, and what I've learned over doing these experiments, is the cost of production of the virus is
Mhm.
very expensive. And to get enough, assuming that we have to use that 2.2 number per kilogram, it can cost up to $500,000 to make enough virus to put it into a patient. Now, the beauty is it would be then presumably last the lifetime, but it's very expensive, and technology is rapidly evolving though. So by the time this is ready to, in 2 years, who knows, they might have that cost down by, twofold, fourfold, who knows? So it'd be more reasonable.
Can I ask an additional question? In some of the therapies that are already on the market for, I wouldn't say gene therapy, but, for the other enzyme replacement therapies that have already been on the market for, similar conditions like PKU, usually patients have to take another medication to fight off any reaction that they might have. Would there be a similar something with gene therapy that they would have to have an administration, just because, you're introducing, I know it's a virus, but that has this gene encoded in it, but just so that the body didn't,
Reject it.
Yeah.
The answer is, I don't know for humans. In mice, we didn't introduce any additional thing.
Mhm.
And we don't see any indication that the mice are mounting an immune response against the virus or the CBS protein. So in the first study we did, the one that didn't work very well, one reason we thought it might not work well was because the mice were producing antibodies, but we did not detect any antibodies that cross-reacted. So I don't think, well, and then the second experiment, of course, worked real well. So I don't think it had to do with I don't think that was it. But mice are not humans, and in humans, it could be a problem. Ron would be a better person to answer that.
Oh, no, that's fine. It's interesting, and it's all very exciting, to be at this point in time. I remind people that our last FDA approved drug was now about 25 years ago, to kind of see with the light speed that things are taking off. It's really exciting. To think that gene therapy could potentially be a cure. It's like a unicorn. You have the idea of what it is, but it's this mythological thing. And that was the same way with the word cure. We had the concept of it, but it just seemed like this out of reach thing that was never really going to exist.
Right. And I think that's changed a lot now. And
yeah.
We're going to see, I predict especially this next decade, we're going to see more and more of these rare diseases will be treated by gene therapy, and especially rare diseases where the liver is the primary organ.
Yeah. So there's a couple more questions here. Do you anticipate there are going to be age limitations on a trial for gene therapy when we do get there?
Yeah, that is a very good question. I have a feeling that the very first, what we call dose escalation studies, will probably restrict that to adults, just because nobody wants to accidentally, if there is some bad thing that happens, we don't,
Mhm.
But I don't know. It's a very good question. I mean, some of my view on this is that we're doing it in pretty young mice, and so ultimately I think the most benefit would be the younger you do it, the better.
Yeah. And I think that's probably kind of following the progression of what we see in other therapy development areas, is they start with adults and then they work younger. And I think you make a good point, is that no one wants to put a child or an infant in harm's way, especially since they can't make those decisions for themselves, whereas adults, we can make those decisions. And that's, I mean, it definitely, though, once we know it's safe and effective to be able to start it in children, it does, because then they have that lifetime of potentially knowing nothing else, that it's just, I have this condition, but I've had this treatment and I'm cured.
Right. One thing I would say is, in my experiences, and this might be unique to the population in Qatar, but when I went to visit the group so Qatar, is this country in the Middle East that has very high rate of homocystinuria. They have about 150 patients just in this little country of 2 million people. And the physician who's kind of in charge of treating all these patients, he said the real issue is when they're little, the parents do a great job of controlling their protein intake because they can control their diets. It's when they become young men or women and they're teenagers, then it becomes really hard because they want to socialize. So much of socialization is around food that they are not as compliant as they need to be. I think that's the perfect age, frankly, for a trial. Like a nice teenagers, I think would be the ones to do it. It's just me.
I think that problem is universal after talking to so many parents, just because teenage years are about independence. There's one last question here I want to squeeze in before we close out here. And do you think that these trials would first roll out in the US, or do you think they would roll out in Europe?
I think they're going to roll out in the U.S.,
Okay.
but they could be both. I'm not sure about that.
Okay.
But my hope is that would be in the U.S. But we'll see. As I said, the talks are ongoing, and that's a whole nother issue because although I'm the scientist, I don't actually. The patent is owned by the institute, so they're the ones that will have to make the final negotiation. And yet, whenever there's a negotiation, they get a zillion questions and they say, Warren, what are the answers to all these questions? so I have a little bit of say. So hopefully, though, my goal, and I'm going to be and Ron's goal as well, is one way or another, we're going to do our very best to. I would like to see a human study initiated soon as possible, because I'm very optimistic about it. And frankly, for my, selfish scientific desires, I would feel really good to know that something I worked on helped somebody lead a better life. So
I don't think that's selfish. I think, that's what your life's work has been really dedicated to. So,
but most scientists don't get to make that, you're building on work, and your work gets built on by others, and just to be able to see it is just would be a treat.
Yes. Well, thank you so much for your time this morning, and sharing your expertise and knowledge with us. It's such an exciting time for the homocystinuria community, just being able to see how quickly it's advancing now. We can't wait to see where your work goes. And I'm sure that, we're going to be seeing the fruits of it here within the next few years. And as as your gene therapy continues to progress, we'll make sure to keep the community up to date, and we'll probably be inviting you back as it makes those next steps.
Well, it'd be a pleasure.
Yes. And so we do have one more, our part 2 of our summer mini series. They're back to back today. And so that part 2 takes place at 10 p.m., not 10 p.m. That would be a little late, 10 a.m. Central Time. So in 30 minutes after a brief intermission, there's an opportunity to learn about patient registries for the homocystinuria community, natural history studies, and the 2 current clinical trials, one including is one that Dr. Kruger had mentioned at the start of his presentation with Travere, and another company called the GLIA. So the registration is still open to that if you would like to join us and you are not registered. So we'll hope you'll be back. And again, thank you, Dr. Kruger, for your time and expertise this morning. We greatly appreciate it, and we can't wait to hear back from you in the future.
Thank you.
All right. Have a great rest of your day.